Hello everyone, welcome back to one more video session. In this session, I'm going to talk about the Mendel's first law, that is law of dominance, and also second law, that is law of segregation. And even I'm going to talk about the test cross as well as back cross. All four concepts are very important actually, so I just want to enclose them within one video. Okay, so I told you that in 1900, a scientist called as the Carl Coren actually, he modulated Mendel's general rules based on his plant hybridization experiments, and he just gave a name to his each rule. The first rule, he just renamed it as law of dominance. For the second rule, he gave a name, that is law of segregation, and the third rule, that is the law of independent assortment. I like that actually. The Carl Coren actually modulated the rules and gave a name actually like this. Okay, so I'm going to talk about Mendel's first law. Now we can consider it as Mendel's first law, that is law of dominance. Second law is law of segregation. The law of dominance and law of segregation both are related to Mendel's monohybrid cross experiment. Please remember this. Whereas the law of independent assortment is related to Mendel's dihybrid cross experiment. So entirely different. The first two laws comes within monohybrid cross experiments. Okay. See, within that one law actually you could see three important sentences actually which is formulated by the Mendel itself. So you have to learn all three. If the question is like this, define Mendel's law of dominance, you have to write all these three sentences. It's very simple, very easy. Okay, so characters are controlled by discrete units called as factors, and then factors occurs in pairs, and then actually in a dissimilar pair of factors, one member of the pair dominates the other. You know about it already because you know about the monohybrid cross experiment. Okay, so for example, if we take this genes, actually a pair of genes, actually we call it as alleles. So allele number 1 and allele number 2. Okay, they always occur together. Okay, obviously, right? See here the characters. For example, let us take character, that is the height of the plant. Correct. So if we consider height of the plant, obviously we know that there are two contrasting traits. One is actually tall character, the other one is the dwarf character. For the tall character, we need this type of alleles or genotype, or we need this type of alleles or genotypes. Correct. So that is actually, for one character we need, or it is controlled by discrete units. See here, you could see unit in the sense here, nothing but alleles. Okay, or you can also write genes. So that's it. Okay, and these units are called as the factors. That is according to Mendel. Now also we call factors as genes itself. We don't use the word that is factor. We just call it as gene. So capital letter T is one gene, and this is actually another gene. And these two genes for a character, let us take tall character, that means actually capital T, or it could be capital T, small t. For dwarf character, it is always recessive genes, actually small t. They always occur in pairs, correct, right? Yes. If we consider any character, that is the height of the plant, actually there are two types, as I said. Obviously, that particular character is controlled by discrete units which always occur in pairs, correct? See, you cannot simply write T, capital T. Correct. If only you consider only capital T, obviously the character will not be expressed. You need either another dominant gene or either another recessive gene. So always they occur in pair. Okay, so this is one thing. If they doesn't occur in pair, obviously the characters will not be expressed. This is very important. Even though there is no function in this particular genotype, the tall is there, that is capital letter T is there, obviously the plant will be taller because that is the dominant gene, whereas small letter T is the recessive gene. It will not be expressed because it is suppressed by capital letter T actually. Okay, but still they have to be in pairs, otherwise the plant will not express any characters, either not even tall or not even dwarf characters. Remember it. The third one, in a dissimilar pair of factors, very simple actually, we can consider the F1 generation of monohybrid cross. That means we considered the height of the plant, that is tall and dwarf. Okay, due to the crossing between homozygous tall and homozygous dwarf, that is capital T into small t, we got in the first generation, that is heterozygous tall plant, correct? So it is a dissimilar pair of factors, correct? See, one is capital letter T, other one is small letter t. So it is a dissimilar pair of factors. Out of dissimilar pair of factors, obviously the plant is heterozygous tall because this capital letter T, that is one gene out of two dissimilar pair of factors, dominates. the other dominates small t. As a result, small t cannot be expressed. Instead of that, the capital T dominates and it gets expressed. Okay, as a result, the plant, the phenotype will be tall itself. Okay, so in a dissimilar pair of factors, one member, that's what I said, like one member, only one capital letter T, that is one member, dominates the other, that is the recessive one. So one member of the pair dominates, called as the dominant gene or dominant gene. Okay, and in this case it is the recessive gene. The dominant gene in dissimilar pair, let us just consider capital letter T, and the recessive gene is small t. Doesn't matter, this one will dominate, whereas this one get non-expressed, okay, unexpressed. That is what actually, that's all about the law of dominance. So you have to learn this and you have to write as it is, don't alter this. Got it? Okay, so this is actually a law, you have to write like this. So this is actually law of dominance. Now I am going to talk about law of segregation. So now let us just learn about Mendel's second law of monohybrid cross experiment, that is the law of segregation. According to this law, it says that the two alleles received, one from each parent, they segregate independently in gamete formation, so that each gamete receives one or the other with equal probability. Okay, so let us see that first. The two alleles received. So we take one example here, that is the F1 generation. I am going to say the two alleles. I am going to write that two alleles here. So I am going to consider the heterozygous tall plant from F1 generation. So you know that the two alleles received, one from each parent. So we have two alleles here. This is actually, let us say, one allele and this is the other allele. The two alleles are present within one organism, which is a heterozygous tall plant. Here, out of two alleles, the one allele is given by the parent. So let us say this, correct. So that particular one allele is given by the parent. Then let us take this as male and this is the female, and same thing, the another allele is received by another parent. So ultimately it finally combined during the zygote formation because of the fertilization process. That's what I said, the two alleles received, one from each parent. Okay, one from each parent, we have it now, which is now in a hybrid, actually in a heterozygous plant. Okay, so this is for, let us say, this is paternal allele, this is actually the maternal allele, given by the mother, given by the father. During the gamete formation, so I'm going to write, see what happened actually. So here they segregate independently in gamete formation. What is the meaning of segregate? Segregate means they split, they separate, they separate. Okay, that means two alleles now separate. Let us just assume that there are two gamete formation possibility. Let us just assume like that. Okay, so during this, so one gamete will receive capital T, the other will receive small t, correct. So let us just name it, this is gamete 1, this is gamete 2, correct. So for this gamete 1, capital T is there because it is, these two actually separated during gamete formation. This is gametogenesis. During gamete formation, capital T entered here, whereas small t entered into the another gamete. This is one possibility. Even you can also like assume that this one, this one go to the gamete 2. That is also there, correct. Okay, capital T may go into gamete 2, whereas small t can go into gamete 1. But remember that in the gamete, obviously, we will not see a pair of alleles, obviously, right? Any one should be there, that's it, because it is haploid, this is diploid, that's what I actually said. So that each gamete receives one or the other, that's what. Each gamete receives any one, this one, allele actually, okay, with equal probability, that's it. There is a 50% chance. One thing to remember that during the formation of the gamete, there is no mixing up of the alleles takes place. You will not see any gametes with this kind of alleles, correct? Obviously, right? You will not see this, correct? So there is no mixing. Obviously, they separate during gamete formation. So we call that there is no blending. Please remember this word. There is no blending in the sense there is no mixing up of the alleles during gamete formation. They split out. That's what actually the law says. Okay, so hope you understood the first law, that is the law of dominance and the law of segregation. Please remember this. No need of to remember all these things, just you have to remember the law. Okay, it may be asked. test cross is one of the very important concept. It could be asked for your board examination and for NEET also. For example, what is the phenotypic ratio of a test cross? First of all, let us just learn what is exactly test cross. Okay, so we know that let us just assume that by taking a different example, same, that is monohybrid cross itself, but I am taking a different character this time. So let us assume that a homozygous violet colored plant, like the flower, okay, violet colored flower is crossed with the homozygous, that is recessive white color, because actually here this W, I have written both capital letter W, that is actually for violet color, okay, or purple color, whereas the recessive character is the white. Okay, so we have taken two contrasting traits here, that is violet, which is dominant, whereas white is actually recessive. Okay, so due to the cross, you know that what you have to write here. So the gamete, one possibility is here capital letter W. Obviously, I am not going to write another gamete because it is homozygous. Obviously, it will have the same type of allele. Okay, here we write small w. Okay, so now these two fuses during fertilization process, and as a result, you know what you get is heterozygous. That means whatever the plant which are produced in F1 or filial generation number 1, which are all violet colored flower, correct? Because of the presence of the capital W, that is according to Mendel's first law of dominance, correct? Okay, so this will suppress this one, that's we know it. But when you are doing the experiment, you may not know what is the genotype. You don't know what is the genotype of this organism, correct? Phenotypically, that is, the phenotype is simple. Actually, this is violet, this is white. As a result of the cross between them, you get violet colored flower plants, obviously, but you don't know what is the gene which is present inside this, correct? So, to know this genotype, to know the unknown genotype of the F1, what we can do is, we can simply just cross this with its recessive parent. Obviously the parents are two. Let us consider this is a male and this is female, or female or male parent, whatever it is. Okay, so this is actually the offspring. This offspring which is obtained in the F1 can be crossed with the recessive parent, not with the dominant one. Okay, so for example, we can do that now. What we do is okay, we split like this. Here I am going to write, let us assume that, for example, we don't know, there are two possibilities to get a violet colored flower. It could be WW, or it could be capital W, small w. Whatever it is, obviously the phenotype will be same actually for both different types of genotypes. I don't know about it, but I will consider two now. Okay, so what I am going to do is, first I will consider the homozygous dominant. Here I am taking F1 as homozygous dominant because I don't know the genotype. Simply I consider F1 as homozygous dominant. So what I have to write here, that is capital letter W and capital letter W. Okay, and I am going to cross it with its parent, particularly the recessive parent. Okay, that is the test cross. So I am going to write small w, small w. Okay, so we just draw the Punnett square here. Okay, so now let us just write this particular organism's genotype here, that is the gamete W, capital W outside, and this one's here. Okay, small w, just cross it, you get W, small w, and capital letter W and small w, and here capital letter W and small w, and finally again capital letter W and small w. All the offsprings which is produced due to the cross between that homozygous dominant, homozygous violet colored plant or flower plant with the recessive parent, obviously all the plants which is obtained during F2 generation, all are violet, correct? So all are violet. So we did not get any recessive plant here, correct? We did not get any white flower plant here. That means we can conclude that the F1 could be this, that is homozygous dominant. Okay, but in this case actually we don't know the genotype, but we got violet colored flower plant actually. So now let us assume we take another possibility. Okay, so what we are going to do is, again here homozygous, not homozygous, here I am going to just write heterozygous dominant. Okay, heterozygous dominant here. So I will take capital letter W and small letter W. This is heterozygous genotype for violet color because W is there. Obviously, it is dominant. Okay, it is crossed with its recessive parent. Okay, so here what I'm going to do is, again I'll do the same thing. So very simple now. Okay, so W here, small W here, small white here, small white here, and the cross here. W, okay, W and small W. Here we have got small w and small w, and we have small w, again we have small w. So that means if you look into the phenotype, 50% of the plants, particularly out of 4 offsprings, the 2 are showing the the violet character, that is violet flower, whereas 50%, that is 2 out of 4 showing the white flower character. Correct? So that means if you look into the phenotype, so what is the phenotype here? So phenotype is very simple. So 2 violet colored flower and 2 white colored flower actually. So the phenotype can be 1:1. That's it. So please remember the phenotypic ratio for test cross is 1:1. Here you don't get it because all the flowers or offsprings are which are violet, that is dominant. So that means actually we can come to the conclusion that this is homozygous WW. In this case, as we got recessive, see, we got exactly 50% of the recessive, same like as the parent. That means we can come to the conclusion that yes, this is heterozygous. Okay, so what is now test cross? Okay, please write on here the phenotypic ratio 1:1 and also write 50% plants are violet flower, and again 50% of the plants have white flower. That's it. Here all the flowers are violet. Okay, so simple actually. So you have to write these two for the test cross if it is asked in the examination. Very simple actually. So what is the definition of the test cross? Very simple. An organism, right here, organism is, we better write dominant. The organism which is dominant actually, obviously. We don't know the genotype, but we know the phenotype. That is actually the color is violet, the violet flower. Obviously, it could be capital W or capital W, small w. There are two possibilities. An organism showing the dominant character is crossed with its recessive parent. Recessive parent. Why it is crossed with recessive parent? Simple. We know that white colored flowered plants obviously having homozygous small w. That's it. Okay. So an organism with a dominant character is crossed with its recessive parent. That is called as the test cross. What is the significance of test cross? See, finally we got the answer actually. Very simple. To know the genotype of the unknown organism, unknown plant or unknown organism. That's it. To know the genotype of the unknown organism, because obviously in the beginning we did not know what is exactly the genotype of this violet colored plant. It could be W, that is capital W small w, or capital W. We did not know, but now we came to know that yes, due to the monohybrid cross between homozygous violet and homozygous white, we got heterozygous violet. Correct? So you could see here. That's it. Okay. We can say that now, yes, F1 plants are heterozygous violet flowered plants. Correct? That's it. So that is actually the test cross. You can also write like, to know the genotype of the test organism. Which is the test organism? The test organism is F1 generated or generation plants actually. That is actually the test organism. So that's all about the test cross. Now I'll just tell you about the back cross. That is not required. Maybe like for NEET examination. What is back cross? Back cross is very simple actually. To get the pure line, the back crossing is very, very important. Simple. This F1 generation, that is actually a particular F1 generated the generation plants which are crossed with recessive parent or with the dominant or homozygous parent. So that means actually you can select any of the parent. Okay, there is no restriction, but the backcross means that particular organism, the F1 organism, should be crossed with either with the mother or either with the father. That's very important actually. Why? Ultimately to get the pure line actually. Okay, so that is one thing, to to eliminate any foreign genes if it is there within the F1 generation. That's it. Okay, but in the case of test cross, it is entirely different. Ultimately, to know the unknown genotype of this organism, we have to cross with only recessive, not with this dominant. Okay, for example, if you get a question like, so F1 plants are crossed with homozygous plants, then what type of cross it is? 100% it is not test cross, it should be back cross. Correct? The test cross means always recessive and F1, whereas back cross is always F1 with this one, the homozygous, or the homozygous recessive, homozygous dominant or homozygous recessive. That's very, very important actually. So please remember back cross. So I'll write here back cross, F1 with father or mother, any of the parent. But test cross is 100% F1 with its recessive parent. We don't know whether it is mother or father, but 100% it's recessive parent. Recessive parent. So that is actually the test cross. Hope you understood this. Okay, so that's all about it. So as homework, I want to give you some questions, particularly from monohybrid cross as well as from test cross. Okay, so let me see like your understanding level.